How are you? How are you all?
Good, sir. And you?
I'm fine. I'm well. I was not well a couple of days ago. I'm well today. I got cold. Okay, so any doubts before I start? Anything you want to ask? Let me just share the notebook. Okay, can everyone see the notebook that I'm sharing?
Yes, sir.
Yeah. So in the last class, we finished with electron transport chain, photosystems, photosynthesis, the basic. And I asked you to go and read about Kelvin Cycle C3 and C4 plants, because in today's class, we'll be starting with the process of biosynthesis of starch or glucose, sugar basically, which is known as the carbon fixing. So we are fixing CO2 now. So we have reached the stage where we are ready to fix the CO2. This is what we do in photosynthesis, correct? In order to make the food. Correct or not? So let me just give you a brief overview of what all things we have. So we have all the recipes ready. So we first studied about how oxygen was figured out to be a crucial part of this process through Joseph Priestley's experiment. And then also we figured out that glucose, like Julius von Sack said, that glucose is produced when plants grow. And he also figured out that chlorophyll, which is present in the chloroplast, is a crucial requirement for that to happen, which means that oxygen and glucose are the players which are there. Now we needed to, the third one is sunlight, and the fourth one is water. So for sunlight, basically Engelmann figured out that a white sunlight, if you scatter it into its constituent Vibgyor pattern, then each color has its particular wavelength, and because of a particular wavelength, it has a particular energy associated with it, correct? And he showed that the rate of photosynthesis is more in the blue and the red wavelength of the sunlight through this aerobic bacteria's accumulation experiment. Is it clear? So we got sunlight also in the picture. And about this chlorophyll, the pigment, I also told you about chlorophyll A, which is the primary pigment, then carotenoids, xanthophylls, and chlorophyll B is also present as accessory or secondary pigments. And all this graph was clear to you that how rate of photosynthesis actually follows the similar kind of graph as of the absorption by these pigments. Okay. Then the fourth thing required is water, because water is required along with CO2 to make glucose. Now, this was the overall reaction for photosynthesis. And I told you that we also discussed what is the difference between plastids, chloroplasts, thylakoids, and chlorophyll. So all of that is clear to you. Then I went on to, we started with the two types of reactions in photosynthesis. So basically we have all the ingredients ready. We also know by now that there are two different kinds of reactions which takes place. So basically they are, one is light dependent and the other is not light dependent. So the light dependent one is called the light reactions or the photochemical reactions. And the other one conventionally was called dark reaction, whereas simply it's just carbonic or carbon reactions, or you say synthesis reactions, okay, where you synthesize the sugar. Now, then we went on to understand the photosystems, the LHC, reaction center of chlorophyll A. And there are two kinds of photosystems, photosystem 2 and photosystem 1, and how the energy which is trapped from sunlight is captured by the first, like the LHC of the second PS2, photosystem 2, at a lower wavelength and a higher energy, and then how this electron excites to a excited state, gets accepted by electron acceptor, and in order to come back to the ground state, it has to give out that energy, and that energy is utilized in the electron transport system to make ATPs from ADP, right? And then further on, when electrons from PS2 comes to ground state, still they are like in between, they are captured by the LHCs of the, like that energy is captured by the, so sorry, the electrons are passed over to the LHC of the PS1, and again it gets accepted by electron acceptor, and in the second term this electron which was just being passed on is now utilized to make NADPH, right? And I told you that ATP and NADPH are two types of high energy rich chemical intermediates, correct? So all of this is clear till now?
Yes, sir.
Yes, we also studied that for the first LHC, which is of the photosystem 2, the constant supply of electron comes from the splitting of water, right? When water splits, it gives two water molecules splits to give 4 electrons, and those electrons are constantly fed to the LHC system. And then if any system, if any plant lacks the PS2 system and only has a PS1 system, then it has to go into a kind of cyclic photophosphorylation process where there is only one LHC of PS1. So water is not required to feed that LHC with electrons now, because that electron is in a cyclic reaction, constantly making ATPs, and no NADPH is synthesized. Therefore, there is no NADP reductase also when PS2 is lacking, okay? So the first one was the non-cyclic photophosphorylation. The second one is the cyclic photophosphorylation. And then we talked about the position. And I ask you, with all these raw ingredients that we have now ready, and all the processes that we know that can happen in the thylakoid membrane, now we have to figure out that whatever happened during the electron transport chain and this photophosphorylation, we made ATPs and NADPH as a product of the light dependent reactions, correct? So at the end of light reactions, basically what we have harvested from the energy from sunlight is these two molecules, ATP and NADPH, correct? So this is the story till now. Everyone on the same page with me? If anyone has any doubts, please ask before I move forward. All of you are on the same page with me? Say yes or no?
Yes.
Okay. So what is the next obvious question? So we have ATP, NADPH ready. We have understood the half of the reaction, which is a light reaction. And now we have high energy rich molecules with us in the system. And clearly, we also know what is the final goal we have to achieve. We have to make food. We means plants have to make food finally. So till the half part in the interval, they have made ATP and NADPH. So they are not food yet, right? In order to make food, which is like starch or glucose, they have to reduce CO2, correct? So obviously, the next question is, what is the role of these ATP? So where are ATPs and NADPH used? Okay, what do they do? And how does CO2 comes into picture? And if CO2 comes into the system in order to get reduced, there should be a molecule or a chemical reaction happening. So there should be a molecule that will basically accept this CO2, correct? So you have to take CO2 and you have to reduce it. It cannot get reduced on its own, correct?
Yes, sir.
Yes. So these are the two next obvious questions that we have to understand today. Now I asked you to go and read about C3 and C4 pathway and Kelvin cycle. Anyone got to know about what is C3, C4, Kelvin cycle before I start?
Yes, sir.
Yes, Mariam. Which one will you talk about first?
C3.
What is C3?
Whose first product of CO2 fixation is C3 acid?
Yes, a 3-carbon acid. C3 acid basically means an acid with a 3 carbon. Okay, C3 acid is not an acid actually. We call it C3 pathway because what you said is the definition of it, that the first product of the reduction of CO2, first stable product, has to be a 3 carbon acid, right? So that's a C3 pathway. So through both the pathways, basically what we will do is we will reduce carbon dioxide into starch. Okay. So there are two types of pathways, C3. And what is C4?
Whose first product is OAA, oxalo something acid.
Yes, which means whose first product, first stable product will be 4 carbon acid, right? A 4 carbon product. So in C3 pathway, we have a 3 carbon product, which is the first stable product of that, when the process of reducing this CO2 starts. And in the C4 pathway, the first stable product is OAA, oxaloacetic acid, which is a 4 carbon. You're right. Now, Kelvin cycle is basically, some things are very basic. So when I told you about, let's go up and let me give an example to make you understand. So this Z scheme, which also has the electron transport system in it, where this whole electron is being transported and finally utilized. So this Z scheme is the non-cyclic photophosphorylation itself, correct? Because it's non-cyclic and its photophosphorylation is happening in order to make ATP from ADP. This phosphorus is being added. Am I correct?
Yes, sir.
Yes. So the Z scheme itself is a non-cyclic photophosphorylation process. In order for it to be cyclic, you just have to lose one photosystem, which is photosystem 2. And the only way now the system can survive or can continue to exist is evolving a cyclic photophosphorylation, which is this pathway, which only has PS1, right? So basically non-cyclic phosphorylation is not something different. It is just the Z scheme, correct? Yeah, you're following me? Similarly, when we talk about C3, C4 pathways and Kelvin cycle, this C3 pathway is basically, Kelvin was the person who figured it out. So Kelvin cycle actually takes this, the 3 carbon molecule, which is in the C3 pathway, it gets fixed through Kelvin cycle. So Kelvin cycle is a part of both C4 and C3. Do you understand? So C3
Yes,
is
sir.
something different, but just the Kelvin cycle itself. In order to evolve a C4 pathway, we need something extra that I will tell you about now. And clearly, this ATP and this NADPH, which are high energy chemical intermediates, will be used in these pathways. So these high energy molecules will be used in C3 and C4. Is it correct? Pathways. So you understood the purpose of ATP and NADPH?
Yes, sir.
Now, there is still one question left, which is this CO2. Now we need CO2 as a raw material, as a recipe. Now we got it. So which will be the first molecule to accept CO2? Okay. Now, normally, let me tell you this, that we knew that in C3 cycle, you can write this down, in C3 pathway, the first stable product of CO2 fixation, the first stable product of CO2 fixation is a 3 carbon acid, C3 acid, which is known as PGA. phosphoglyceric acid or phosphoglycerate, PGA. Okay, this is the 3-carbon acid, which is the first stable product of C3 pathway. Now we knew this information, and we knew that CO2 is, taken and reduced. Now to reduce CO2, if the first intermediate is a 3-carbon molecule, PGA is the first stable intermediate and CO2 is the first raw material, then how many carbons does CO2 have?
1.
So 1 carbon we already have. And we knew that the first stable product is a 3-carbon molecule. So what will you think that the molecule which accepts this CO2, how many carbons will that molecule be having?
2.
Right? 2-carbon molecule should be accepting. So this was the general thought, that a 2-carbon molecule must be accepting a CO2 in order to make a 3-carbon stable product, correct? This is what you will also think logically.
Yes, sir.
But this happened to be wrong. This was wrong. And the molecule that accepts CO2, like the CO2 acceptor, instead was RUBP, which is a 5-carbon molecule. Now this made things a little bit complex, correct? Now there's a confusion. We have a 5-carbon molecule accepting a CO2, then it will become a 6-carbon molecule, and finally the stable product is a 3-carbon molecule. So it's not just taking CO2 and reducing it. Whatever is the final product, it's also splitting up, right? It's also breaking down. Make sense? Because if a 5-carbon molecule, RUBP, write down the full form of RUBP, it's called ribulose 1,5-bisphosphate. And this is our entrance question also. The CO2 acceptor, ribulose 1,5-bisphosphate. Okay, hello Aisha. Aisha has joined a little late. It's fine, it's fine. How are you?
Fine, sir.
Okay, we are just started, okay? So I was just telling that a CO2, which is a 1-carbon molecule, we knew that it's a 1-carbon molecule, we know. We also got to know that the first pathway that got discovered was the C3 pathway, where it is called a C3 pathway because the first stable product of the CO2 fixation is a 3-carbon acid, which is called PGA, phosphoglyceric acid. This PGA is a 3-carbon molecule. CO2 coming in is a 1-carbon molecule. So scientists normally thought that a 2-carbon molecule must be a CO2 acceptor. But that was proven to be wrong. Instead, the CO2 acceptor was RUBP, which is ribulose 1,5-bisphosphate, which is a 5-carbon molecule. Now from here things got complicated, and then Kelvin started the experiments to understand this whole pathway, and the cycle hence was called Kelvin cycle. Is it clear? Is it clear, everyone?
Yes, sir.
Now write down the Kelvin cycle was discovered by the Kelvin cycle. So we'll study Kelvin cycle. The Kelvin cycle was discovered by Melvin Kelvin and Benson. I think his name was Andy Benson. So Melvin Kelvin and Andy Benson together figured out this Kelvin cycle, elucidated this Kelvin cycle. And write down the final carbohydrate product, formed directly from the Kelvin cycle. Like when the Kelvin cycle ends, the final product is not a glucose. Okay, so the final carbohydrate product formed directly from the Kelvin cycle is actually not glucose, but a 3-carbon sugar. Which is called, so the final product, now pay attention to this. The final product of Kelvin cycle is called G3P. Okay, its full form is glyceraldehyde 3-phosphate. Is it clear, everyone?
Yes, sir.
So this is the final product that comes out of Kelvin cycle. Okay, glyceraldehyde 3-phosphate, 3GP. It's a 3-carbon molecule. Now let's try to figure out. Then what happens is, now if a 3-carbon molecule comes out as a final product, and you have to finally make glucose at the end of photosynthesis, then there must be additional steps to it, correct? Right? So let's first figure out, write down this one thing. The Kelvin cycle reactions can be divided into 3 parts. So we'll study step by step. First, the Kelvin cycle. The Kelvin cycle reactions can be divided into 3 parts. First part is carboxylation. Second part is reduction. And third part, because it's a cycle, if anything is a cycle, you have to come back to the first initial product, correct? Am I correct? Like in a cyclic photophosphorylation, the end product should be the electron, which should come to the LHC to again become the first initial thing to begin with, right?
Yes, sir.
Yes. So if you do carboxylation, reduction, that finally in the end you will have to do regeneration. Third part is regeneration of RUBP. So this Kelvin pathway managed to end at the same place from where it began, this RUBP. RUBP accepts a CO2, goes through the same whole pathway, then finally comes and becomes RUBP to accept another CO2. Do you understand? So this got clear when we figured out that RUBP is again made. So clearly, logically, this is obvious that all this RUBP is doing is taking a carbon and fixing it, correct? Fixing it to make a 3-carbon molecule product or finally a glucose. Is it clear?
Yes, sir.
So this Kelvin cycle is also a part of carbon fixation cycle. Do you understand how and why? Okay, do you understand how and why? Because we are taking CO2 as raw material. So basically,
Yes, sir.
plants are doing, they are fixing carbon from atmosphere to make starch finally, Okay, and Kelvin cycle is the first step of it. Now write down. Let me first make the cycle and let's understand the cycle, then we'll go into the details of it. Okay, shall we do that? Let me use black. So let's try to figure out Kelvin cycle. Okay, now we all know that it starts with, let it start with RUBP. Okay, ribulose 1,5 bisphosphate. Is it clear? And we know that this RUBP takes, what it does, it takes from atmosphere? Tell me.
CO2.
One CO, like let's take CO2. So to begin with, there are 3 molecules of RUBP from where the reaction starts, and 3 molecules of CO2. 1 molecule of RUBP takes 1 molecule of CO2. Is it clear?
Yes, sir.
Now, this is the, okay, let me not take 3 in the beginning, otherwise you will get confused. Let's start with 1 only. So when RUBP takes CO2, we all know that the first stable product is going to be a 3 carbon molecule, because Kelvin cycle is itself basically a C3 cycle, correct? So it will make a 3 carbon stable molecule, which is 3 phosphoglyceric acid, PGA, correct?
Yes, sir.
This 3 is a part of the molecule. You can also just write PGA. You can also write the full form, which is phospho. And remember, I told you that any acid, when it is inside the system, it is not in the acidic form, correct? In any cell, if there is acid inside it, it is going to harm the cell. So what do cells do to store acid? How do they store acid?
They make acid.
Carbonic acid. In our, when I was teaching you human physiology, carbonic acid, how it was in the, in which form it was in the blood? Was it bicarbonate ions?
Yes.
Negative, right? So similarly, this phosphoglyceric acid stays in the form of phosphoglycerate. Okay. 8. The word 8 comes, which means it loses a hydrogen and loses its acidic form. Otherwise, it's going to destroy the cell. So this is the first stable product, which says that it's a C3 pathway. Is it clear, everyone?
Yes, sir.
Then this phosphoglycerate now becomes 1,3 bisphosphoglycerate. Bisphosphoglycerate. Now you know that this 3 or this 1,3 is the position of the what do we say it? This phospho group, right? So if there is one,
Yes, sir.
it is at the 3rd carbon. It's called 3-phosphoglycerate. If there are 2 phospho groups, it's on the 1st and the 3rd carbon, bisphosphoglycerate. Is it clear? Now, to do this, to add something, to add a phosphorus, energy is required or not? We will require energy, correct?
Yes, sir.
So here, this is the first step where ATP is utilized. Okay, and specifically, okay, I will come to the numbers later. ATP is utilized, and what does comes out? ADP plus inorganic phosphorus. Clear? So the ATP that we made during the light reactions first gets utilized here, correct? And whenever, this is the rule of thumb, remember this, whenever you use energy in any cyclic pathway, in any cyclic biosynthetic pathway, whether you are making glucose, you are making anything in the body, if you commit energy to it, it is non-reversible. It cannot go backwards. Do you understand?
Yes, sir.
Because if this 1,3-bisphosphoglycerate can go backwards and make 3-phosphoglycerate back, then this energy utilization is waste. And body never wants to waste energy. So whenever you commit ATPs in any pathway, that step becomes, and this is also asked in entrances, that step basically becomes irreversible. So this irreversibly becomes 1,3-bisphosphoglycerate. Clear?
Yes, sir.
Now this 1,6 this part where we are adding CO2 to RUBP is known as carboxylation part. Is it clear? Because we are adding carbon dioxide. So this part is carboxylation part. Clear, everyone? The first part that I talked to you about. Now write down about carboxylation one more thing. Okay. Or should I just complete the cycle first and then go and dictate you things? I think
Yes.
that's right. Yeah. After carboxylation, once this irreversible product 1,3-bisphosphoglycerate is formed, from here onwards, we need to reduce it. So the reduction part starts. Okay. And how do we reduce it? Remember, apart from ATP, what did we also make? As a
NADPH.
yes. And what do NADPH has a lot to give? H, hydrogen, right? So now we will utilize NADPH again. It will come and donate the hydrogen and itself will become NADP plus, correct? And when it will donate, let me go a little. And when it will donate hydrogen, this 1,3-bisphosphoglycerate becomes glyceraldehyde 3-phosphate. What does this becomes? Glyceraldehyde 3-phosphate. Remember this molecule, this name? I just dictated
Yes,
something about it above. The final product of Kelvin cycle is this, glyceraldehyde 3-phosphate here. So after reduction, we reach the final product of Kelvin cycle, correct? Now this glyceraldehyde, this glyceraldehyde 3-phosphate can go out of this cycle, or this glyceraldehyde 3-phosphate, can continue the cycle, where again I will write glyceraldehyde 3-phosphate. This G3P can continue in the cycle, and this phase now where it's continuing is called regeneration. And here what happens is we use again ATP, like we use energy. So if ATP is used, ADP plus inorganic phosphate comes out. That's the thumb of rule. And this energy is utilized in regenerating. So this G3P, it is a glyceraldehyde 3-phosphate. From here, we have to again make it into RUBP, which goes back, and this whole cycle starts. Is it clear? Do you understand, everyone? The basic cycle is clear.
yes, sir.
Now, here I told you that this glyceraldehyde 3-phosphate has two options. Now, if all the glyceraldehyde 3-phosphate leaves out of the system of the Kelvin cycle, then clearly the Kelvin cycle will shut down, right? So it has to go into the regeneration phase. Now that depends. Now I'm using black to just balance things, like how many molecules come and participate. So to begin with, there are always 3 RuBP molecules which take on 3 CO2 molecules. Do you understand? Now things will start making sense to you. So if we take 3 RuBP and 3 CO2 and we have to make a 3 carbon molecule, how many 3 PGAs will be made? How many these molecules will be made?
2.
Sorry?
2.
No, not 2. It's a 3 carbon molecule. RuBP itself is 5 carbon molecule.
Greater than 3?
It is 3 RuBP, and each RuBP is each RuBP is a 5 carbon molecule, remember?
Yes, sir.
So don't get confused. That is the most confusing part you have to. That's why I'm going slow, so that you understand it. I'm not hurrying up, because if you understand this process, then everything in plant systems, plant biology is, less confusing than this part. RuBP is ribulose 1,5-bisphosphate, which clearly tells that there are 2 phosphate groups. One is the one on the carbon 1 and other on the carbon 5. So at least it has 5 carbons. So, and it has 5 carbon molecules. Ribose sugar, the word ribulose or ribose, remember there is a ribose sugar in DNA also. 5 carbon sugar in DNA, right? That's ribose sugar. So it is a 5 carbon molecule, and we are using 3 RuBPs. So how many carbons did we get from RuBP only? 5 into 3. 5 3s are
15.
And from CO2 we got 3 carbons. How many carbons in total? 15 plus 3. What's difficult in this maths?
18.
18. So how many 3 carbon molecules, 3 PGA we can make? If you have 18 carbons and you want to make a 3 carbon molecule, how many such molecules can you make? People?
6.
6. So we have 6 into 3 PGA. Is it clear to everyone?
Yes, sir.
Now, if we have 6 such molecules and each molecule uses 1 ATP, then how many ATPs we are going to utilize? 6. Correct? And similarly, how many ADPs will come out? 6. And when we, after this ATP addition, sorry, this PI will not come out. I'm so sorry. I forgot. Why this PI will not come out? Can anyone tell me? It's a silly mistake.
Because we are adding phosphate.
Yes. So that one PI is getting added. It's becoming bisphosphoglycerate, na? But 6 ADPs will come out. That's clear. Now, how many 1,3-bisphosphoglycerate molecules will be made? 6 only, because there were 6 3-GPAs to which phosphate was added to make 6 1,3-bisphosphoglycerate. Clear? Everything making sense till here? And how does this 6 came? Because RuBP itself is a 5 carbon molecule. Let me write it here. It's a 5 carbon molecule. Everyone on the same page with me till now?
Yes, sir.
So how many NADPH will you need to reduce 6 molecules of 1,3-bisphosphoglycerate? 6.
6.
And how many NADP plus will come out?
6.
Now we have 6 molecules of, यहाँ पे भी 6 होगा, because we have 6 molecules of glyceraldehyde 3-phosphate, correct? Everything is 6 till now, correct everyone?
Yes, sir.
From which only 1 molecule of G3P, glyceraldehyde 3-phosphate, goes out in one cycle. Is it clear? That's the trick. That's the catch. That is how biology has evolved. So only one molecule is allowed to leave, because when one molecule will leave, how many carbons will leave? Come on. How many carbon will leave? 3. 3 carbon will leave the system, correct? G3P has 3 carbons, correct?
Yes, sir.
Yes. So 3 carbons will leave the system. How many carbons are left?
3.
Oh, there were total, how many carbons we started with?
3.
15 plus 3, 18 carbon we started with. Till now, everywhere there are 18 carbons. It's balanced. 6 into 3 PGA, again, 6 3s are 18 carbons. 6 into 1 3 bisphosphoglycerate. This molecule also is 3 carbon molecule. So 18 carbon. 6 into G3P, again, 6 3s are 18 carbon, out of which one G3P molecule is allowed to leave. It took 3 carbon with it. How many
15.
carbons?
15.
Which means how many molecules of G3P left? 5.
5.
Right? So 5 molecules of G3P goes back to follow the regeneration, because 5 molecules of G3P means how many carbons? 15. And how many carbons are there in one RUBP?
5.
5. So how many RUBP we can regenerate back? How many?
3.
3. See na? And these 3 RUBPs goes again, which is this 3 RUBP. So the cycle has to be maintained, correct? So in each cycle, basically what are we doing is we are only allowing 3 carbon molecules to exit the cycle in the form of glyceraldehyde 3-phosphate, which is the final product of Kelvin cycle. Is it clear, everyone?
Yes, sir.
Now look here. If there are 5 molecules of G3P, we will not use 5 molecules here because of ATP, because this ATP is used to make RUBP, correct? And how many RUBPs are being made?
3.
So there will be 3 molecules as ATP used, and as a result, this PI will not be there. Okay. ADP. And how many ADPs will come out? 3 only. Now this cycle is balanced and complete, correct? So this phase from here till here is carboxylation, till here is reduction, and this is regeneration. Is it clear? Now
Yes, sir.
make the cycle, then I'll. Is everyone crystal clear with this whole cycle? This is called Kelvin cycle. Any doubts in terms of numbers of molecules? Because the question, entrance question, very, very trivial entrance questions are how many C3 first molecules of phosphoglycerate are there in 5 cycles of Kelvin cycle. So you should know how many are there in 1, which is 6. They will also ask how many total ATPs and NADPH are used in the whole cycle. Okay, in one cycle of Kelvin cycle, and to make one glucose. So we'll talk about that later. But this is clear?
Yes, sir.
Make it. Everyone make this cycle. Okay, then go a little back. Sorry. Yes. I think the cycle is visible, right? Tell me when you are done. So I'll move ahead.
Done, sir.
Everyone done? Now, this first step here, let me use another color. This first step here where RUBP is taking CO2, this RUBP plus CO2, the carboxylation. So there is a reaction for this step, which is a very important reaction. So I'm writing the reaction for carboxylation step. So it's RUBP plus CO2 to make what? 3 PGA, right? 3 phosphoglyceric acid. Is it clear? And we know that it's 3 RUBP plus 3 CO2 to give rise to 6 3 PGAs, correct? The most important part of this, please excuse me, this reaction is that there is an enzyme which plays a which plays a crucial role to make this RUBP accept CO2. And that enzyme is called Rubisco. And this enzyme is the most abundant enzyme on the planet Earth. Can anyone tell me why? Rubisco is the most abundant enzyme present on planet Earth because plants are present everywhere. Photosynthetic plants are present everywhere, right from grasses to plants. Around us, if we can say that something is dominating the planet, then it's not animal species, it's the plant species, right? And right from algae onwards, they do photosynthesis. And the first step of Kelvin cycle, which is common both in C3 and C4 type of plants. So whichever plant it is on planet, it must be having Rubisco enzyme in it. And that also not in trace amounts, but in every single cell which can photosynthesize and which has a green pigment, correct?
Yes, sir.
Rubisco is the most abundant enzyme in the ecosystem, or what we call the biosphere of Earth. Write down, Rubisco is the pacemaker enzyme of the Kelvin cycle. Rubisco is the pacemaker enzyme of the Kelvin cycle and the most abundant enzyme of biological system on Earth. Okay, because Kelvin cycle is common for all plants, C3 or C4. So is it clear, everyone?
Yes, sir.
Now you have written carboxylation. I think you don't need to know the reactions for reduction and regeneration, but because they are very structure heavy. Do you want me to give you the reactions for reduction and regeneration? Or is it fine?
Fine, sir.
Okay, so this is the Kelvin cycle. Now this one G3P, glyceraldehyde 3-phosphate, which leaves the Kelvin cycle, what happens to this one, correct? So write down. The final product of the Kelvin cycle, G3P, also known as triose phosphate. Common name. Triose phosphate is just telling that it's a triose means 3 carbon sugar with a phosphate. Okay, you know, triose, pentose, hexose, all these terms.
Yes, sir.
Triose phosphate goes into the starch synthesis pathway. Goes into the starch synthesis pathway. So this triose phosphate, or you can also write, it acts as a precursor for synthesis of starch and sucrose. This final product acts as a precursor for the synthesis of starch or sucrose. Is it clear, everyone?
Yes, sir.
Now let's talk about sucrose synthesis. Now Remember when I taught you about the structure of chloroplast, what big granules it had in the chloroplast? Starch granules. Remember? Should I just go up? Where was it? This one. Can you see here are starch granules? So starch synthesis happens in this chloroplast. Chloroplast is again a cell organelle, but outside the chloroplast is the cytoplasm, correct?
Yes, sir.
So inside the cytoplasm, or we call it cytosol, there is sucrose synthesis. Is it clear? So sucrose is, write down just this one line. Sucrose synthesis happens in cytosol, and starch synthesis happens in chloroplast in plants. Now from here onwards, because this was your Kelvin cycle, and also clearly it's the C3 cycle, correct? Because the first stable product is a third 3 carbon molecule. So this cycle itself is the C3 cycle. Is it clear, everyone?
Yes, sir.
Write down Kelvin cycle is a C3 pathway, which means it happens in all the plants. So if a plant is called a C3 plant, it obviously has a C3 pathway, which is the Kelvin cycle itself. If a plant is called a C4 plant, then what is the difference? So write down, okay, before going there, I think, write down one more thing. Okay. Where should we start it? Now tell me, for every CO2 which is entering the cycle, okay, there are 3 CO2s here which are entering, but for every CO2 which is entering the cycle, how many ATPs are utilized? For every CO2 entering the cycle, Kelvin cycle, this is the question. How many ATP molecules are utilized? These are some questions, just write it down. 3 ATP molecules are utilized per CO2 entering the Kelvin cycle. Write down. You know how 3?
3 CO2.
3 ATP molecules are utilized per CO2 molecule entering the Kelvin cycle. Because there are 3 CO2 entering, hence here are 6 ATP utilized. If there was 1, then 6 by 3, which is 2. So 2 ATPs are used here, and 3 by 3, 1 ATP is used here, correct? Is it clear?
Yes, sir.
So total 3 ATPs are used per CO2 entering the Kelvin cycle. And how many NADPH are used? 2. 2 NADPH are used per CO2 entering the Kelvin cycle. Write down that also. Now, to make a molecule of glucose, I'll tell you about it later, but just write down. To make 1 molecule of glucose, how many CO2s are required? How many CO2s are required?
Sir, to make 1 molecule of
Glucose. Glucose is a 6 carbon molecule. So how many CO2s are required? 6. Simple. I told you we have already written the complete reaction of photosynthesis. Where is it? Where is it? Where is it? Where is the complete reaction of photosynthesis? We did write it, right?
Yes, sir.
Yes. So 6 CO2 is required to make 1 molecule of glucose, correct? Which means 6 times this cycle has to go. 6 Kelvin cycles are required, correct? So if 6 Kelvin cycles are required, how many, the question is, how many total ATP and NADPH molecules are used to make 1 molecule of glucose? This is also an entrance question. Just right now, you wrote how many ATPs and NADPH are used for 1 CO2 entering the cycle. Now I'm telling you there has to be 6 such cycles happening where 6 CO2s turn by turn has to be fixed. So the whole cycle has to go 6 times, which means to make 1 glucose. So if the question, the twist the question, it comes like this: how many ATPs and NADPH molecules are utilized to make 1 molecule of glucose? Then what will be your answer?
24.
18 ATPs. 18 ATPs and 12 NADPH. Write down. For 6 CO2s, write down, to make 1 molecule of glucose, 18 ATPs and 12 NADPH. It's clear, na? See, while explaining you this, I just balance the whole reaction. I balance the whole cycle. And when I balance the whole cycle, I told you that the cycle started with 3 RUBP taking on 3 CO2. I am telling you 6 CO2s are required. So whatever is happening in this one cycle, one balance cycle, is actually happening in 3 terms, right? To balance it, I have just written it 3 here. Do you understand what I'm saying?
Yes, sir.
And to make 6 CO2s come into the cycle, whatever is happening here, so we are here using 6 plus 3, 9 ATPs and 6 NADPH. We will just double it. So 18 ATPs are utilized and 12 NADPH are utilized. And if they ask you how many ADPs come out and how many NADP plus come out, then you will write 18 ADPs come out and 12 NADP plus comes out. Clear? Or NADP?
Yes, sir.
Say yes or no. Is it clear to you, everyone? Any confusion in this?
No, sir.
No. Now, the question is, to make a glucose molecule, how many triose phosphates are required?
2.
One is a 3 carbon molecule. So 2 will be a 6 carbon molecule. Okay. Done?
Yes, sir.
Very good. So now write down C4 pathway. The C4 pathway. Let's go to C4 and see what's different in C4. C3 and Kelvin and the number of molecules going in, out, everything is clear to you, everyone. You have to remember this cycle by heart. Believe me. So I just explained you to the best I could. Now let's go to C4. Now we already had a C3 pathway. So why there was a need of a C4 pathway? What can you think must be a reason? What is lagging in the C3 pathway? Then you need a C4 pathway. Can anyone think of anything? Why do we need a C4 pathway? Okay, just think about it and give me 1 minute. Okay, I'll be just coming back. I'm asking, the question is, why do you think C4 has evolved? Or do you think C4 is primitive or C3 primitive? And if you think C4 is like advanced, then why is the need of C4? What do you think is happening in C4? Or what is lacking in C3 or Kelvin cycle? Okay, just give me 1 second. I'm coming back. Yes. Hello. Hello, everyone. Hello.
Yes, sir.
Yes. I'm back. Uh, throat is still a little sore. Okay. So anyone came up with any brilliant idea? Think as a plant. You are a plant. Vegetable.
Sir, because of environment,
Because of some?
habitat.
Yes, you are correct. Evolutionarily, that was one of the reasons that drove it. But if I ask you what kind of environment?
Desert,
Desert.
high intensity of light and
You mean a more like a dry tropical region, correct?
yes, sir.
Yes, you are correct. It's the dry tropical region, and we have a very big dry tropical region. You know which is the tropical region? Tropical, subtropical, tundra region, temperate region. You know about these geographically?
Sir, I don't remember.
From equator, when we go towards the pole, we go from the hot tropic,
Yes, sir.
to temperate and then to correct. So we have a big tropical region. We have big lot of plants. So write down few things about C4 cycle first. C4 is an alternate mode of carbon fixation. C4 pathway is an alternate mode of carbon fixation that has evolved to minimize photorespiration, and optimize the Kelvin cycle. To minimize photorespiration, I'll tell you what photorespiration is. Let me first. To minimize photorespiration and optimize the Kelvin cycle. Okay. Now write on next point. Plants which are adapted to dry tropical regions. Plants which are adapted to dry tropical region have evolved C4 pathway. Have evolved the C4 pathway. Now the point is, you all know by definition, also write down, in the C4 pathway, or you can also write, it's called a C4 pathway because the first stable product of CO2 fixation, the first stable product of CO2 fixation is a 4-carbon acid, 4-carbon molecule, or a 4-carbon, just write 4-carbon molecule, C4 molecule called OAA, oxaloacetic acid. Oxaloacetic acid. Is it clear?
Yes, sir.
Now let's see how it is better or how it is different. If not better, then it has to be different. See, things can evolve for two reasons in biology. One, you need something better. Okay. And most of the time you need something better when environment changes or something changes, or in the same environment when life becomes a little more competitive or different, correct? So you need to do something different. So you evolve something different, correct? Which is more efficient or something like that. Or you need to make something different. You understand? So both are equally good, but both are different things. Do you understand the difference between something better and something different? Something better clearly means that it is better over the previous version. So over time in evolution, what is inferior gets lost or is not, like, it cannot sustain. But if you just are diverging, if you are making something different, if you're evolving something different, then both of them can stay. For example, lungs and gills. You cannot say that lungs are better than gills, right? They are something entirely different. Gills are still there in aquatic organisms like fish. Fish cannot have lungs, right? And we cannot have gills. Is it clear?
Yes, sir.
But if you compare between other organs, let's say kidneys, heart, or brain, and we go across evolution, then lower organisms do not have better good brains. Like their brains are very, very primitive, correct? We have well-developed brain. So that is called making something better. And that better has made us the arc rulers of the planet because we are very, very intelligent beings. Is it clear? In this case, this C4 pathway that has developed, it has developed because of different anatomy of the plants also. So plants are different. So now they can utilize their different anatomy and can have a different, slightly different pathway that becomes more optimized, which can optimize the Kelvin cycle and do better. Now, how it can do better? Let's see. So let's try to now in the plants, first of all, write down before I go forward, characteristics of C4 plants. Characteristics of C4 plants. So first thing is write down both monocots and dicots. Now you know the difference between monocots and dicots from the last chapter, single cotyledon and double cotyledon, monocots, dicots.
Yes, sir.
Apart from the difference in their cotyledons, there is also difference in their stem structure, the stem and the root structure. Is it clear? And also the difference is there in their leaf structure. So I urge you to go back today and read, because that's important, the difference between monocots and dicots with respect to leaves, stem. We will discuss it. Okay, I'll cover it, but you read it to understand before I started. Roots. Okay. On the basis of cotyledons, I have already told you. You figure out on the basis of leaves, stem, and roots. If any one of you has seen the leaves of wheat plant or rice plant or maize plant, I told you all our cereals are monocots. And if you will, so you cannot clearly, have you seen a corn seed? Popcorn, corn?
Yes, sir.
Can it be split into two? No.
No.
No, that's why it pops. It has water inside, a liquid slide. There is a liquid inside that upon heating pops and the corn becomes popcorn. Okay. If it could have split, it would just have splitted into at heat. So it could not. Similarly, the same thing is with rice, right? Or you can also make same thing of if you put rice in something hot, a hot sand, it also pops like a popcorn. The same thing can happen with wheat and also with bajra and things like that. So it has one cotyledon. So clearly it's monocot. But if you see its stem and roots and leaves, there are long leaves with parallel venation. You know what is parallel venation? What is reticulate venation? All of this is basic stuffs, class 9, 10th. A leaf which is long and have veins like this is called a parallel venation. You will see such veins in grasses, in bamboo, in maize, rice, wheat. And then you have a leaf in which the veins are like this, right? There is a mid vein and there are accessory veins. So this is a parallel venation and this is a reticulate venation. Is it clear? This you will see in mango, guava, higher plants, all your dicots, gram. Is it clear?
Yes.
Yeah. So this is the difference in leaves. About the stem, you will see monocots have slightly hollow stem. Straw. We call them straw. We don't call them stem most of the times. Wheat straw, rice straw. Clear? You understand what I'm trying to say? Also,
Yes.
there's a difference in roots. So you figure out what is the difference. This is your assignment for today. Okay. Now, keeping this aside, let's come to here. So what were you writing? What was the last part you were writing, which I was dictating?
Both monocots and dicots.
Yes. Both monocots and dicots includes in the C4 plants, are included in the C4 plants. You can write examples, corn, sugarcane, and cypressy dicot. Cypressy, corn, sugarcane, and all the plants in cypressy family. Cypressy over here. Cypressy family. So all these plants are your C4 plants. I asked you actually to figure out what are C4 and C3 plants. Some examples. Did you do that? Do that.
Yes.
Okay. If you have not done it, please do it. And write down one more point. These plants, the C4 plants, are adapted to arid climatic conditions. Arid. Anyone can tell me the meaning of arid? Dry, basically drier, where there are two things. In arid climates, the temperature is very high, right? All our tropical around the equator and to some extent, you know, we have a line called Tropic of Cancer and Tropic of Capricorn going through India. No? Which line goes through India? Tropic of Cancer or Tropic of Capricorn? Some geography. Does equator goes through India? No idea. No one knows about the world. Okay. Write down areas which has higher temperature and longer photo period, which means high light intensity, which clearly means area around the equator. Okay. So this is called your arid environment. So these plants are adapted to arid climatic conditions. And we all know that at higher temperatures, the rate of photosynthesis increases or decreases?
Increases.
Decreases. Because I also, okay, I should not have disclosed it. It decreases. You know why it decreases? Because, yes, Mani. Why it decreases?
Less water.
Yes, one thing is if a temperature is high, very good, brilliant. If temperature is very high, you will have less water. So there'll be less things feeding your PS2, right? Less electrons feeding your PS2. But we already have a solution for that. We can just make a cyclic photo system, right? Photophosphorylation, correct? Just have PS1, forget about PS2, you don't need water. So that thing can be, okay, it can be mediated, but your answer is right, but plant already has an answer for that problem, correct? Do you understand, Maria?
Yes.
But you're thinking along the right lines. So let's remove water. Water will be less, but water problem can be tackled by just having PS1 and a cyclic photophosphorylation. We can still make ATPs. What is the other problem?
Very high temperature.
Yes. So what is the problem at high temperature? Do we feel good at high temperatures? Do humans feel good at high temperatures? When we have fever, does our body feels good? Everything is good in our body?
No.
What happens to our metabolism? In fever?
It slows down.
Yes, it decreases, right? Metabolic activities decreases. Our immunity also decreases, correct? But,
Yes, sir.
and it also, I also told you that our body heats up or goes in fever to kill certain pathogens also, right? Certain bacterial infections can be recovered if we have fever. So body for fever for a body is a defense mechanism. But all of this has one thing common. What happens at higher temperature? Why our metabolism goes down? Why we can kill pathogens? Why at higher temperature cells are not functioning properly? Why? Because the functioning of enzymes are inhibited or are disrupted at higher temperatures. Remember, every enzyme has an optimal
Yes, sir.
temperature in which it can work the best. Most of the times it's the body temperature for humans. So if we go at high temperatures, like in a desert or in a tropic African countries, all the plants in African countries will be C3 or C4.
C4.
Yes, C3 cannot survive there. It's clear, because C4 can still do photorespiration and photosynthesis and can produce starch at higher temperature and high light intensity. Is it clear?
Yes, sir.
Write down. They are adapted to arid climatic conditions and are able to give higher photosynthetic output and are able to give higher photosynthetic output at even at high temperatures and high light intensity. So that is why C4 plants are better than C3 plants. But for which condition? The tropical or around the equator conditions. If I go to Europe, then which kind of plants will do good?
C3.
Yes, right. Now do you understand it? So in arid conditions, we have more of C4 plants. Another thing that C4 plants does is they minimize photorespiration. What is photorespiration? Photorespiration. The word says everything, photo and respiration. What is respiration?
Breakdown of
Loss
glucose,
of gases. What do we take in respiration?
oxygen,
Give out
CO2.
CO2. So let's define it. Just write it, photorespiration. You have finished the point that it minimizes photorespiration. C4 plants minimizes photorespiration. And that's why they have more output for photosynthesis. Okay, clear?
Yes, sir.
Now write down photorespiration. Let's also define this. Photorespiration is the process. Photorespiration is the process. Now the word photo clearly tells that this kind of respiration can only happen in the presence of
Sunlight.
light. So it's photo dependent. So photorespiration is the process of light dependent uptake of oxygen. Light dependent uptake of oxygen and release of CO2 from organic compounds. Now how it is different from normal respiration? We do, so for us, for animals to respire, do we need light? Is it light dependent?
No.
But in plants, there is photorespiration, which is light dependent. So write down this difference. The gaseous exchange during photorespiration, write down the gaseous exchange during photorespiration resembles the aerobic respiration. It looks similar. The whole process is similar like aerobic respiration that we do. Resembles aerobic respiration. But photorespiration is a light dependent process and is catalyzed by Rubisco. Make sense, everyone? That's why only plants can do it, because only plants have Rubisco. And that's why Rubisco is not only required in Kelvin cycle, but also in photorespiration. So it's the most abundant enzyme, both by mass and by like its presence. Is it clear?
Yes, sir.
Yes. And who okay. Now, there is something you have to understand. Remember, I told you that plants give out oxygen, okay, as a byproduct of photosynthesis. You all are on the same page with me? See, this is something
Yes, sir.
that I'm asking. Yes. Now try to just first understand my question. Plants give out oxygen as a byproduct of photosynthesis because it's not required in the photosynthesis process. And it was also discovered that more oxygen presence, the presence of more oxygen inside the system inhibits photosynthesis. Do you understand these two terms? If you have more oxygen in the system where photosynthesis is happening, then the photosynthesis will be inhibited. Is it clear?
Yes, sir.
Write down. And it was found by Otto, not the Otto Warburg. So this is an entrance question. I don't think it will be asked in your boards because I don't think it's in your NCERT, but it's important. So Otto Warburg found something which is known as Warburg Effect. So because we are talking about this, I thought, let me tell you about this Warburg Effect. Just write it down. So what did he figure out? He made an observation, write down. Warburg made an observation that presence of O2, oxygen, inhibits photosynthesis in C3 plants. And why it does so? So there was a problem. Now this thing, that when Warburg says that if there'll be more oxygen, this photosynthesis will be reduced in C3 plants, then everyone said that it doesn't make sense. You know, what does oxygen has to do? Anyways, oxygen must be coming into the plant system for respiration process, correct? So he named something as photorespiration, that the respiration is dependent on light. And he also figured out, later it was figured out, that this enzyme Rubisco is not what we thought it is. So we used to think in the beginning that Rubisco is only able to do carboxylation. So whenever Rubisco is present, RUBP will take CO2. Do you understand? And it's true, it does that, but we think that Rubisco only does carboxylation. But later it was found that Rubisco can do both carboxylation and oxygenation of RUBP. Do you understand? So there's a competition. When there is more CO2, when there is more O2, O2 starts competing with CO2 to get added into the RUBP. So Rubisco can do both. It can catalyze both carboxylation and oxygenation. Is it clear, everyone?
Yes, sir.
So that's why if you have more oxygen in the system, that more oxygen, how it will decrease photosynthetic rate in C3 plants, because C3 plants have no other way to fix CO2, and their fixation of CO2 solely depends on Rubisco. And Rubisco, when it sees more oxygen, it can shift from CO2 to O2. So it can, instead of doing carboxylation, can start doing oxygenation of RUBP, and then Kelvin cycle will never start, and then glyceraldehyde 3-phosphate will never be made, and then these two molecules can never be joined to make glucose. So photosynthesis will not happen. Do you understand?
Yes, sir.
So that's why it's important that you tend to remove O2 from the system in a efficient way. Now write down, Rubisco is a bifunctional enzyme. Write down, this is also asked. That's why I told you. Rubisco is a bifunctional enzyme. Okay, I was telling you what is Warburg effect. Sorry. Write down. Oh, I have. Did I tell you that in the presence of oxygen, photosynthesis in C3 plants decreases?
Yes, sir.
Yes, that is Warburg effect. Okay, that when in C3 plants, whenever there is more oxygen availability, the photosynthesis decreases, because Rubisco is a bifunctional enzyme. It can catalyze both carboxylation as well as oxygenation of RUBP. It can catalyze both carboxylation and oxygenation of RUBP. So this is the problem. Now write down. Let's come back to C4. Photorespiration is clear, everyone? Warburg effect is
Yes, sir.
the dual nature of Rubisco is clear. So by affecting Rubisco, you can affect whole photosynthesis, and that's Warburg effect. Now let's come to C4 back. So what did you write in C4? The last thing that you wrote in C4? Tell me. The last line for C4 pathway. What did I ask you to write?
C4 pathway becomes the first stage. Oh, C4 pathway becomes the first stable. That part.
What C4 pathway becomes? Sorry, I could not figure it out. Have you written that plants? Oh, how do we reach that photorespiration? I have to think that. So basically, I was telling you that C4 has evolved to minimize photorespiration, right? This must have been the last sentence. C4 plants have evolved to minimize photorespiration, right? Have you written this?
Yes, sir. I thought the one previous you were saying that one.
Yes, okay, C4 is clear. Now write down, there is one more name for C4, which is, so write down, C4 pathway was discovered by Hatch and Slack in Australia. So it's also called Hatch and Slack pathway. C4 is also called Hatch and Slack pathway. It was discovered in Australia in 1966. Australia. Okay. C3 pathway can also be said just the Kelvin pathway. C4 can also be said Hatch and Slack pathway. Is it clear? Now let's come and see how, let's go and see how C4 do all that, how it reduces photorespiration and increases the rate of photosynthesis even at higher temperatures. So to do that, we had to go to the leaf, the anatomy of C4 plants. That is what I was telling you. So write down the leaf anatomy of C4 plants first. I'm trying to make it as a story. Do you understand everything till now?
Yes, sir.
Yes. So write down leaf anatomy of C4 plants. So C4 plants are unique as they possess. C4 plants are unique as they possess two types of photosynthetic cells. Two types of photosynthetic cells. Okay. So they have an inner ring and an outer ring of cells. Okay. So if you'll see leaf from the top, and this is from the bottom, they have an inner ring, write down, and inner ring of bundle sheath cells. Someone was asking about sheath cells, right? In previous classes, bundle sheath cells. These are the inner cells. They are also photosynthetic. And then there's an outer cell, which I think Mariam was asking, the mesophyll cells. Correct, Mariam?
Yes, sir.
And it was very premature at that stage for me to tell you, otherwise you will just get confused. All plants do not have this, correct? So basically C4 plants have developed two types of photosynthetic cells, bundle sheath cells inner and mesophyll cells outer. Okay. So there is one word that is asked in entrances. I don't think if it's an NCERT. If it's an NCERT, please let me know. It's called Cran's anatomy. So Cran's anatomy is this arrangement of two different types of photosynthetic cells in C4 plants, along with chloroplasts arrangement, which I'm not telling you because it's just because I don't have to define it if it's not coming into boards, but just know that Cran's anatomy is only present in C4 plants. And this is Cran's anatomy. The organization of mesophills in outer layer, then in the inner layer, there is bundle sheath, and there are chloroplasts arrangement, a special type of arrangement, which is called Cran's anatomy. Is it clear?
Yes, sir.
Now, let's go into the cycle itself. So everything is clear. They have a different anatomy. They can do well in arid environments and all that. They have less photorespiration. All is clear. So to make this cycle, we also have to make two different cells. So let's say, okay, let me use different color. Let me use, sorry. Oh. So let's say this space that I'm making here is the outer layer. And which is what is the outer layer called? Mesophyll cell, okay? And this mesophyll cell is connected. So this is a mesophyll cell, and it is connected to the inner layer. And what is the inner layer called? Bundle sheath cell. So please draw it yourself, because while drawing it, it will be less confusing. Now these mesophyll cells and the bundle sheath cells are attached to each other through plasmodesmata. Everyone is clear with plasmodesmata by now?
Yes, sir.
Yes, the direct junctions between cells, okay, through which cytoplasmic streaming can just happen. The apoplastic and the symplastic water movement, remember?
Yes, sir.
Yes. So these are plasmodesmata. They're connected by plasmodesmata. Now C4 cycle starts in the mesophyll cells, and how it is different is that here, rather than Rubisco does not takes in CO2. Instead, CO2, oh, let me use black. Instead, CO2 is taken in, sorry, and the first acceptor molecule of CO2 is your bicarbonate, HCO3 negative. Is it clear? Is it clear?
Yes, sir.
Sorry, CO2 is taken in and converted into bicarbonate. Remember, CO2 can easily be converted into carbonic acid. There is an enzyme called carbonic anhydrase that converts CO2 into bicarbonate and bicarbonate back into CO2 from the blood vascular system in humans.
Yes, sir.
Is it clear, everyone? So here, when CO2 comes in and becomes HCO3 negative, this reaction is done by, so here is an enzyme which is called Pep Carboxylase. So instead of Rubisco, the first enzyme that greets CO2 in this system is your Pep carboxylate. Is it clear? Say yes or no?
Yes, sir.
Now, this Pep, this CO2, now there are same steps. This undergoes, because it's still, if you just convert it into HCO3 negative, it's still a source of carbon. So it has to undergo carboxylation. So the first step is carboxylation step. And as a result of this step, it gets converted into OAA, a C4 acid. Is it clear, everyone? Clear?
Yes, sir.
Now remember, when you did carboxylation and after carboxylation, it's very, when you have very high light intensity and you have mesophyll cells on the outer side, so instead of Rubisco, which is already working in photorespiration and Kelvin cycle, if you ask Rubisco only to go and accept CO2 when it's coming in the outer cells, which is mesophils, then that enzyme's activity or its output is going to decrease. So why it is more efficient than C3 is that it brings in CO2 and asks PEP instead to go and convert it into HCO3 negative, so that we can easily carboxylate it without the use of Rubisco, right? So by this way, you can bring in as much CO2 as you want without burdening the Rubisco system. Do you understand this, people? Say yes or no?
Yes, sir.
Now this OAA, which is a C4, the carboxylated product, now it's a 4 carbon acid. This is transported in the bundle sheath cells. So this C4 acid goes here. Now it's oxaloacetic acid, but inside the cell it will be in the form of oxaloacetate. Is it clear or no?
Yes, sir.
Yes, but I'm still writing it as C4 acid just for the sake of its nomenclature. But remember that it's all the acids inside the cell are in the form of their ates. Now this has to undergo decarboxylation. That's different in C4 system. There was no decarboxylation in C3 system. Remember, there was carboxylation, reduction, and regeneration, right?
Yes, sir.
Here, this C4 acid undergoes decarboxylation. Now, what we achieve by decarboxylating is what we added. We made a 4 carbon molecule. Now we will make it a C3 acid. And what is the C3 acid? What is the C3 acid?
PG. PG.
That is for the C3 system. In C4, that's not the case, okay? But I'll tell you. But just remember that there's a C3 acid. Is it clear?
Yes, sir.
And this C3 acid, which is, yes, you're right, it's a pyruvate. When we decarboxylated, what did we remove? Tell me.
Carbon, CO2.
Yes, perfect. We removed CO2. We took CO2 here. And finally, in the bundle sheath cell, we got a CO2 and we also have a C3 acid. This would not have happened if it was a C3 system. Now let's go to C3 system and try to appreciate it. To make a C3 acid here, 3-PGA, is everyone Paying attention?
Yes, sir.
CO2 came and got utilized. We never got it back, did we? No, right? We never got it back. So it got utilized, and here this CO2 which came out from decarboxylation, can I say that it can now go and get utilized in the Kelvin cycle? Remember, Kelvin cycle is common in both, which is this.
Yes, sir.
So where is Rubisco? So this thing, this whole thing that I have taught you before is happening in C4 also, in the bundle sheath cell. Oh. So I'm making a star here, okay? This is where Kelvin cycle is happening. And the CO2 which we got from outside, we just converted into OAA because we wanted it to be transported in the bundle sheath cell, where we can decarboxylate and get CO2 back. And we can still do it because we have not used any ATP till now, right? So we can get that CO2 back and we can pump all that CO2 into the Kelvin cycle. So one thing that's happening here is Kelvin cycle is happening, correct? So Kelvin cycle is independently happening. What will happen to this C3 acid? This C3 acid is sent back into the mesophyll cell, where pyruvate, where it will undergo regeneration to make what? What should we make? Which enzyme should come back again?
Rubisco.
Pep. Mariam, Rubisco is in here, in this small box. Everything about Kelvin cycle is happening here.
Yes, sir. Yes, sir.
So by using the same CO2, we are doing more work in C4. Do you see? We
Yes, sir.
took in CO2. Instead of Rubisco, we asked Pep to go there, convert it into HCO3, then carboxylate the HCO3 to make it oxaloacetic acid, then transport the oxaloacetic acid in the bundle sheath cell, where again decarboxylate, just reverse the process, take out CO2, pump that CO2 in the Kelvin cycle. So Kelvin cycle is still happening. So everything that we have studied above, this part, C3, this part is happening, correct? And we ensured that this is made, we ensured that glucose is made, we ensured that Rubisco is also made, because Rubisco is there in that part, in this small box. Apart from that, this C3 acid is again pumped back to mesophyll, and through regeneration of its, because this is a separate pathway, C4, this PEP is again made, which is again sent here, and the cycle continues. Do you understand?
Yes, sir.
Instead of 3 steps which were there in C3 pathway, in C4 pathway we have 5 steps. Is this pathway clear to you? Please tell me.
Yes, sir.
Pakka?
Yes, sir.
Okay. So let me also tell you certain things. I'll use a blue color to mark it. So when CO2 enters, Pep carboxylate converts it into oxaloacetic acid. This oxaloacetic acid, okay, does not when crosses the plasmodesmata and goes into the bundle sheath cell. Inside the bundle sheath cell, there is a chloroplast of the bundle sheath cell. There is chloroplast in both, in the mesophyll cell and the bundle sheath cell. Is it clear? And inside the chloroplast of both, inside the chloroplast of bundle sheath cell, this Kelvin cycle is happening. Is it clear, everyone?
Yes,
So if the question comes that in a C4 pathway, where does the process of Kelvin cycle or the process of making synthesizing sucrose or glucose takes place, then you will say the chloroplast of the bundle sheath cell, because they have 2 types of cells. In the chloroplast of the mesophyll cells, there is no Kelvin cycle happening. Is it clear?
Yes, sir.
Okay, very good. So write down C4 pathway involves 5 stages in 2 different cell types. So there are 5 stages in 2 different cell types. The first stage is, a question can also come in the board that list out differences between C3 and C4 pathway. So I think when I'll complete this part now, you will be able to do it. So write down C4 cycle has 5 stages in 2 different cell types. First is fixation of CO2. First is this one, fixation of CO2, sorry, by the carboxylation of Pep, by the carboxylation of Pep. Write down the full form of Pep. Pep is phosphoenol pyruvate. P for phospho, E for enol, next P for pyruvate. So carboxylation of Pep happens with the enzyme Pep Carboxylase, okay? And fixation of CO2 happens by the carboxylation of Pep in the mesophyll cell, to form OAA, to form oxaloacetic acid, which is a 4-carbon acid. This is the first stage. Is it clear, everyone? So I'm writing down, this is number 1 step. Clear?
Yes, sir.
What happens in this step, I have dictated you. Pep comes in the presence of Pep carboxylase, carboxylation of Pep happens, Pep receives CO2 and becomes oxaloacetic acid. First. Second, write down oxaloacetic acid oxaloacetic acid converts into other types of C4 acids. So oxaloacetic acid is one type of C4 acid. So this is the second stage here, where oxaloacetic acid converts into other types of C4 acids. For example, malate and aspartate. So I'm writing it here. It can also become malate. It can also become aspartate. So you are making more options that you can send into the bundle sheath cells. So this is the second stage where oxaloacetic acid converts into other C4 acids like malate and aspartate. Then this is the third part. Third phase is here. We write down transport of the C4 acids. चाहे वो oxaloacetate हो, चाहे malate हो, चाहे aspartate हो. Transport of these C4 acetic acids into the bundle sheath cell. The third stage. Into the bundle sheath cell. Fourth is this one. This is the fourth stage. Decarboxylation of the C4 acids. Now, can you now appreciate that we have multiple sources of different options of C4 acids? In C3 pathway, there is only one particular C3 acid, which is 3-PGA, clear, which is made. But when we make C4 acids, we have options, malate, aspartate. So we are giving more options from which we can take out CO2. So isn't this pathway more efficient? If you have one single thing, so if you say that I will only eat carrots. So if you don't have carrots, probably your system will choke, you will not eat, you will not get nutrition. But if you are okay with eating carrots, peas, mushrooms, everything, then you have more options to get nutrition from, correct? Do you understand or not? It's just about
Yes.
diversity. So a C4 pathway is more diverse because these C4 acids can be malate aspartate, which can be transported into the bundle sheath cell, and then decarboxylation of C4 acids to generate CO2. So this CO2 here is coming from more diverse areas, which enters the C3 cycle. So Kelvin cycle is technically C3 cycle, right? So inside a C4 pathway, there is a C3 cycle independently fitted. So it's better, right? We can now feed this CO2 into the Kelvin cycle. So this is the fourth step. And the fifth step is this one, where we write down, transport the C3 acid, which is left. So the C3 acid which is left is pyruvate. Probably you will like to write it. And when I'm writing pyruvate, you can tell me which is the acid, pyruvic acid, right? In the cell, it is in the form of 8, pyruvate. Clear, everyone? Is it clear or no? Tell me.
Yes, sir.
This pyruvate can be sent back to the mesophyll cell to undergo regeneration and make phosphoenol pyruvate. Now, in order to make phosphoenol pyruvate, we will need energy, correct? From pyruvate to phosphoenol pyruvate, this step requires energy, correct? Say yes or no.
Yes, sir.
So here is a ATP utilized at this step. Okay. And when it goes in the Kelvin cycle, there is the same utilization which we have studied above. But apart from that, an ATP is utilized for this regeneration part. And when this oxaloacetic acid is being converted into many different malate, aspartates, etc., here at step 2, NADPH is utilized. Because we are doing more work, so some more energy can also be given into it. Is it clear? So at step 2 and step 5. Step 2 NADPH, step 5 ATP. Is it clear, everyone?
Yes, sir.
This is your C4 pathway, which has a inside it has a Kelvin pathway of its own. And why it is better? You understood, everything is clear why it is better over the C3 and everything.
Yes, sir.
Very good. Any doubts in C3, C4, Kelvin cycle? Everything that we wanted to finish, we have finished it today. And yes, in time. What perfect timing. So I know that these pathways are a little bit, you have to mug it up or remember, but I tried my best to tell you as a story so that you can understand when you are memorizing it. It makes memorizing easy when you have a story to tell, right?
Yes, sir.
Okay, so please do revise it. C3, C4 pathway, photorespiration, and Kelvin cycle are very important topics for your entrance and also for boards. Okay. In next class, we'll be doing the respiration, like we'll be making glucose, because till now we have only reached till this triose phosphate G3P. We have not made glucose yet. So the pathway we'll make glucose, and then in the next class I will also teach you respiration, because respiration is a topic which I wanted to like the chemical part of it to keep common, because that happens same for plants and animals. So in the next class we have completed everything in photosynthesis. We just have to finish one last step to make glucose, and then I will teach you how to break glucose inside the cell. So till now we have utilized energy, we have utilized NADPH, we have utilized resources, sunlight, water, CO2, and we have made food. So tomorrow I'll teach you what we do with that food. We means both plants and animals do. It's a common thing. Respiration is common. Cellular respiration is common. Okay. Photorespiration was different. Cool.
Yes, sir.
Tomorrow I'll teach you glycolysis, and first we'll make glucose and then we'll break glucose. Okay. Then see you in the next class. If you have any doubts, please ask. If you have no doubts, I declare the class. And please do the assignment, monocots, dicots, and also revise these both C3 and C4 pathways. Any doubts, people?
No, sir.
No doubts? Okay. Then I declare the class. Good night.
Thank you, sir. Bye, sir.
Bye-bye.